Hello everyone, welcome to your lecture on the pathophysiology of glucose regulation. These are the objectives that we will be covering in this lecture. The endocrine system is basically, it affects all aspects of your body. As you can see here, there are a lot of glands, involved in the processes of the endocrine system. It's responsible for growth and development, energy metabolism, muscle and adipose tissue distribution, sexual development, fluid and electrolyte balance, inflammation, and immune responses. Any dysfunction or disturbance in the endocrine system can be categorized as hypofunctioning, hyperfunctioning, and hormone resistance. So hyperfunction is basically when the endocrine gland is absent or impaired, right? And hyperfunction is when the endocrine gland is producing excessive amounts of hormones, right? And then the third thing is when you have hormone resistance. So either receptor, there are receptor defects at the target cells, or their impaired intracellular responsiveness to stimulation by that hormone. Metabolism is categorized as anabolic or catabolic. Basically it's a biochemical reaction. It can be anabolic, where these are growth reactions that promote growth. For example, anabolic steroids, right? They promote growth. People take steroids to build muscle. That's growth, a growth reaction. Then you have catabolic reactions, where the energy reaction caused by breaking down a food for energy. That's catabolic. You're breaking down. So basically, for example, to lose weight, you want to decrease your anabolic reactions and you want to increase your catabolic reactions. Glucose regulation, why is it important? Why is glucose important? Because it's the source of fuel for your brain. Normal cerebral function depends on a continuous supply of glucose from your blood circulation. Now, any severe or prolonged hypoglycemia can actually cause brain dysfunction and even death. That's the reason why hypoglycemia is so important, to understand, to recognize, and to manage immediately. So let's talk about glucose regulation. How does our body regulate glucose? What glands are responsible for this? Basically, when your blood glucose is high, for example, you just had a meal and it's high. Say you ate a large, a lot of, carbs. You ate a big burger, you had like fries on the side, then you had a milkshake, and then later you decided you're going to eat, a huge ice cream or a big piece of cake. So now you have a lot of glucose in your blood, right? So basically, the pancreas are signaled to produce, to release insulin. And this insulin will transport that glucose from your bloodstream to the cells for energy, because that's where the glucose needs to be. It needs to go to your cells so it can use it to create ATP for energy, right? It goes to your muscles for muscle glycogen, right? And it also goes to your liver, where it is stored as glycogen, right? The glucose in the bloodstream, after this happens, the glucose in the bloodstream is decreased and the glucose in the cells for energy is increased. So this process is called glycogenesis. Now, this process also signals the liver to stop the release of glucose, right? Because remember, your liver stores this glucose as glycogen. So it tells the liver, hey, we don't need any more glucose to be sent out into the blood. Stop it. Save it for when we really need it, right? And it enhances the storage of dietary fat in the adipose tissue. It accelerates the transport of amino acids from your dietary protein into the cells and inhibits the breakdown of the stored glucose, protein, and fat. For example, like we said, when we eat a meal, blood sugar increases, pancreas release insulin, the sugar is transported into cells for energy, and liver to be stored as glycogen. Now, what happens when your blood sugar is low? Say you haven't eaten for a long time, you just, woke up after like sleeping 10 hours, which you can't do while you're nursing school, I'm sure. But say you didn't eat for a long time, right? Eight hours pass, you haven't eaten, your blood sugar is low. Now your body is worried. Okay, how are we going to give the brain the glucose that it needs to work, right? So then what happens in that case? The stored glycogen that's in the liver is broken apart into glucose and it's released into the bloodstream, right, as glucagon. This causes a decreased glucose in liver cells. So now you have less amount of glucose in the liver cells, and you have a higher amount of glucose in the bloodstream. So it releases it as glucagon. Right, this process is called glycogenolysis. For example, when you do not eat, like we said, when you don't eat for a prolonged time, the glycogen stored in the liver is broken down into glucagon and it's released into the bloodstream so your brain can get the glucose that it needs. Now, another process that can occur is gluconeogenesis. Now, this occurs when you have 8 to 12 hours without food. The liver forms glucose from the breakdown of the non-carbohydrate substances, such as amino acids. This over here is a basic concept map on glucose regulation. Please review this concept map, and we will talk a lot more about this when we go over the PowerPoint on diabetes. What is diabetes? It's basically a group of metabolic diseases categorized as hyperglycemia, an elevated level of glucose in the blood. Now, this is when you have consistently high levels of glucose in the blood. Because remember, when you have high levels, what happens? The insulin is produced by the pancreas, released by the pancreas, pushed into cells, muscles, adipose tissues, and the liver. But sometimes, your glands don't work as they're supposed to. For example, you have hypofunction or you have resistance, right? As we mentioned, can happen with, endocrine function. So diabetes occurs when you have a consistently high levels of glucose in your blood. That's hyperglycemia, right? There are a lot of risk factors, and we will cover these risk factors even more when we talk about diabetes. But if you notice here, age 30 years, or older for type 2 and less than 30 years for type 1 diabetes. Then you have high density lipoprotein levels less than, that means you don't have enough high density lipoproteins, right? And then you have, high levels of triglycerides or cholesterol levels. You had a history of gestational diabetes or delivery of a baby over 9 pounds is an indication of possible diabetes in the future. Hypertension can also cause diabetes, right? Family history of diabetes. Obesity is the leading cause of diabetes, where you have body weight or BMI of greater than 30 is a huge risk factors for diabetes. If you were previously identified with impaired fasting glucose, impaired glucose tolerance, right? That's also a risk factor. Race and ethnicity is a risk factor. For example, African Americans, Hispanic Americans, Native Americans, Asian Americans, and Pacific Islanders are specifically at very high risk for, developing diabetes. Now, there are different classifications for diabetes. You have type 1, type 2, diabetes associated with other syndromes, also known as secondary diabetes. Then you have gestational diabetes and pre-diabetes. This chart over here gives you a lot more detail. Please review this before you come to class so we can, talk more about diabetes and maybe work on some case studies and questions to understand it better. Okay, we're almost there. Next up will be your lecture on type 1 and type 2 diabetes. Okay, I'll see you in class.